Let's see if I can spell her name. Because at the time it was like, oh, I'm going to Old Town to get margaritas and eat like this. Saturday night. But we don't want to miss some competition. Actually, I've always felt that way. All the trouble. Yeah, exactly. I mean, you might need to break. Like, I feel frustrated I can't do this, but like, but you're not. I'm not going to alternate. Yeah. Right. Exactly. Right. So I was saying to somebody today, like, oh my God, I stay in the sun and I save clothes to my shirt. I am the same color as my shirt. I actually brought this good sunscreen that I've been stretching out because they don't sell it. So I've been stretching my sunscreen. I have it, and I decided I don't need to do anything out of it. I have it for tomorrow where I'm going to sit by the pool. I was sitting by the pool yesterday. Get them in water. It sounds divine. But if it's like this tomorrow, then I'm not going to be doing it. I mean, always a lunch break. I was done. So I didn't think I'd do that. Oh, so do you have
Eventually, they will have to.
One sort of after.
Did you guys just grab the whole row?
Yeah, I don't know.
No wonder. Oh God, this is a terrible view of me. All of my menopausal liver. Thank you very much. Yeah, very sorry. Yeah, great. All right. It is 7:45 and we're going to get started. So I'd like to welcome you. Artyom Kopp and I are the session chairs for plenary session 3. And Denise Montel is also going to be speaking at the very beginning of it. We have a lot of awards to celebrate this evening, and we're going to start off with the poster awards. So there were 3 undergraduate poster presenters and 3 graduates. So we'll start with the undergraduate. So in first place is Abby Matt from WashU. Are you here? Abby may not be back from dinner yet. So great job, Abby. In second place is Grace Woods from Lewis and Clark College. Are you here, Grace? Congratulations. Okay. In third place is Jennifer McIntyre from the University of California, Irvine. Okay, so I think we're going to move to the graduate awards. So in first place is Arya Vaikakara-Chitran from the University of British Columbia. They probably all went to dinner together. In second place, Marian Jans from the University of Osnabrück in Germany. And in third place is Menglin Lee from the University of California, Santa Barbara. Right. From one of the Montel labsies. Okay, and
Wait, is this one of our winners?
yes.
Yes, please come stand.
Oh, Maren Jens, yay, congratulations, yay. If you know any of the awardees, please let them know that they've won. And now we're turning this over to Denise.
Great. Well, good evening, everyone. I hope you're enjoying gathering together at this conference as much as I am. I'm Denise Montel still, and past president of the GSA, which is why I'm here to present the GSA Awards this evening. So each year, GSA presents 6 prestigious society awards in recognition of outstanding contributions to genetic research and education made by extraordinary scientists in our community. And on behalf of the Society, I have the privilege to recognize 4 of the 2022 awardees who are from the fly community. So first of all, let's see, I'm supposed to open a presentation. Let me do that. Do you have to do that? Yep, thank you. There you go. Great. So we have these 6 awards, and 4 of them are going to members of our Drosophila community. So Harmit Malik is being honored with the Edward Novitski Prize for his innovative This prize is for his innovative studies at the interface of evolution and chromosome biology, developing the concept of centromere drive, and his contributions to understanding genetic conflict, wherein competition between genes and opposing functions drives evolutionary change. Harmit couldn't be with us tonight, but we send our virtual congratulations out to him. Next, we have Margaret Fuller, is awarded the Genetic Society of America Medal in honor of her pioneering work on cellular mechanisms that underlie Drosophila spermatogenesis, which led to numerous discoveries pertaining to stem cell regulation, germline-soma interactions, and germ cell differentiation. She's also recognized for her contributions in training and mentoring graduate and postdoctoral researchers. Minx, can you please come up and accept your award?
Yes, please.
What happened was that a group of my former postdocs and graduate students got together, banded together, and made the nomination. And that means so much to me. I feel like if I think back on my largest contributions to science, it's the people that worked in my lab and have gone on and are carrying on their own careers and having their own students and postdocs. Thank you. Thank you all.
Great. Okay. Alana O'Reilly and Dana Ruiz-Whalen are jointly receiving the Elizabeth Jones Award for Excellence in Education, recognizing their work to develop educational programs, amazing programs that promote equity and safe spaces for students to integrate life experiences and culture with scientific ideas while addressing fundamental questions in genetics. Thank you, Alana and Dara, for the impactful work you continue to do. Please come up and accept your awards. So in closing, I'd like to bring your attention to the upcoming and very exciting GSA Awards seminars featuring our, I'm going rogue without the mask here, sorry. Featuring our 2022 awardees, which will be held throughout the month of May. The 2021 series was a huge success, and we're looking forward to hearing more from our esteemed colleagues on their impactful and inspiring work. And so with that, please join me in congratulating Margaret Fuller, Harmit Malik, Alana O'Reilly, and Dara Ruiz-Whalen.
And now on to the first speaker of the evening, Nick Baker. He's going to talk about cell competition.
Okay, thank you. Can everybody hear me? All right, I'd like to thank the organizers for inviting me to give this presentation. I've been attending this meeting since 1987, so I certainly appreciate what a big honor it is to be asked to speak. I'd also like to thank them for bringing me to San Diego. My wife sent me a video this morning of hail falling on our home in New York. So I'm very happy to be here. I'm going to talk about minute mutations, which were described 100 years ago by Bridges and Morgan as mutations with short, thin bristles and a developmental delay. And then in the 1980s, I nearly got an extra hour to speak to you because Erica hasn't turned the timer on yet. In the 1980s, it became clear that nearly all of these, nearly all minute mutations corresponded to ribosomal protein mutations. So after many years studying problems in developmental genetics, what would motivate me to become interested in ribosomal proteins? And I assure you, it's not something that I could ever have anticipated. But I hope that tonight I will be able to persuade you that it's possible to learn some interesting things about development from studies of ribosomal components. So while we were studying growth and proliferation in the marginal discs, we found that antibodies against activated caspases could be useful reagents to study cell death in Drosophila. And that enabled us to look at cell death in imaginal discs mosaic for minute cells and see what you see here. And what you see here is apoptotic death of minute cells, largely where they're in contact with wild type cells that are replacing them. And this is the phenomenon of cell competition, whereby one population of cells eliminates and replaces another population of cells and spreads through the entire tissue. And this is just visualizing what had been described in the 1970s by Murata and Rapal, who showed that clones of minute cells in imaginal discs grew for a while and then disappeared. And there are quite a number of examples of cell competition, genotypes that compete in Drosophila now. I've probably heard of several examples at this meeting. And there are also many examples of competitive phenomena in mammals. Sorry, I keep flipping, I'm not touching it. Many examples of competitive phenomena in mammals. And so it's a very interesting phenomenon, and we decided that we would study it in fruit flies. And we're interested in what are the molecular mechanisms that enable this to happen? And what is the physiological function of cell competition? Why does it happen? And could it be exploited in regenerative medicine? And so we set up a genetic screen to find mutations that would affect cell competition. We exploited the eyeless flip system to make mosaic eyes, so we can make eyes that are 50% red and 50% white cells. And then we used a deletion of an X-linked ribosomal protein gene. It happened to be RPL36. And we rescued that gene with transgenes that we inserted on the autosomes. And then using the flip FRT system, we could generate clones that lose the rescue transgene and are functionally minute. And those clones don't survive in the adult eye because they're eliminated by cell competition. And then we could screen for chemically induced mutations on the autosomes that would allow those RPL36 heterozygous cells to survive. And we discovered quite a few mutations that affected growth by doing this, and a very small number of mutations that apparently had no other phenotype. So they appear to be specific for cell competition. And of course, those are the interesting ones. And this is an example here of a mutation that was isolated by my graduate student, Wei Li. This is a photograph of a current photograph of Wei that I lifted from his lab's website at Penn State. So at the time we isolated this mutation, we really did not in Drosophila have ways to map and identify point mutants known from a single allele. And it was particularly difficult in the case of a mutation that had no phenotype. And so I'm afraid that I forbade Wei from working on this gene further. But we kept the stock. And many years later, another graduate student, Abhijit, succeeded in mapping and identifying it. And he had to use methods that had not been used in Drosophila before in order to do that. And he found that this mutation corresponded to a missense mutation in another ribosomal protein, RPS12. Now, I don't know if you can appreciate how disappointed we were waiting years to try and identify this gene that specifically affected cell composition and identifying a mutation in another ribosomal protein. So here is RPS12, and here's RPL36 on the backside of the other subunit, not anywhere near one another. You know, we had no idea how mutations in these two ribosomal proteins might interact. And we had no idea if this was even something we should be interested in. And we also had no idea how we would interrogate this system. And in fact, we could very easily have dropped the project here. Had it not been, we probably would have done if poor Abhijit's other project had worked. But because all the other things he did ran into problems, periodically he had to return to this problem and try to think, how could we find ways to study this system? And so over time, he did develop ways to interrogate this system. And one of the things that he found was that this RPS12 mutation was not specifically interacting with the RPL36 mutation. It would suppress competition mediated by any ribosomal protein mutant. So here is an imaginal disc from an animal that's entirely RPS18 mutant. And when you generate clones of the RPS12 mutation, they grow as nice large clones, but the twin spot, which doesn't have the suppressive mutation, is lost. So the RPS12 mutant is affecting the competitiveness of apparently all ribosomal protein mutations. And then he did what is the most important single experiment in his whole paper. And this is an important experiment, partly because it involves no mutant genes. All he did in this experiment is manipulate the copy number of wild-type genes. And so manipulated the copy number of RPS12 with transgenes from 2 to 4, and that doesn't make any difference. That doesn't affect growth. But if you do the same experiment in the background of a minute mutation, then clones of cells that have fewer copies of RPS12 always survive, and clones of cells that have more copies of RPS12 always disappear, showing that the wild-type RPS12 gene has a function that promotes cell competitive elimination of cells. If you do the same experiment where the extra copies of RPS12 have this missense allele, then they have no effect. So this mutant of RPS12 has lost this pro-competition function. And so these results led us to think that perhaps all the minute mutations are actually behaving like gain-of-function mutants in RPS12. And if that was really true, we should be able to mimic minutes by overexpressing RPS12. So we've never been able to do that because the Drosophila RPS12 gene autoregulates very efficiently, and we haven't been able to overexpress it. That turns out not to be true in mammals. So we've knocked out the mouse RPS12 gene, and it's haploinsufficient. And indeed, if you express the human RPS12 gene in Drosophila, you mimic the minute phenotype. So we think then, as this was the first evidence, the minute phenotype was not simply caused by depletion of ribosomes, but it represented a specific pathway that was being activated in these cells. And we still don't know molecularly what RPS12 is doing. It may be involved in some kind of specialized ribosome. It may have an extra ribosomal function. We do, however, know what its downstream effector is. And that's because of another mutation that specifically affected cell competition and rescued minute cells. And so when my student Chang was mapping this mutation, he rapidly developed the idea that it was probably caused by this premature stop codon in this putative BZIP domain transcription factor. Now, I didn't believe a word of this because how could the phenotype of a ribosomal protein mutant depend on a transcription factor? But I was wrong, and he was right. This is the gene that's responsible for the rescue. And what happens is that in minute cells, induce expression of this transcription factor in an RPS12 dependent fashion. And this transcription factor actually mediates many of the phenotypes of minute mutants. So it's responsible for most of the developmental delay of minute flies, which is reversed if the transcription factor is mutated. And even the reduced translation that is typical of minute mutants is restored if this transcription factor is removed. So the majority of the minute phenotype is actually transcriptional in origin. So we thought that in order to gain insight into what XRP1 was doing, we would look at how it's affecting translation. And we rapidly found that minute cells contain high levels of phosphorylated EF2 alpha. So the EF2 complex is a translation initiation factor that recruits the methionine tRNA to the small subunit and enables it to start scanning for start codons. And it's a well-known site of translational regulation. So for example, in the unfolded protein response, a kinase called PERK phosphorylates EF2 alpha and shuts down the majority of cellular translation. And indeed, it's the same kinase that's active in minute cells. So that PERK is responsible for the elevated EF2 alpha phosphorylation in minute wing discs, and it's also responsible for the reduction in translation. So we wanted to know if this reduction in translation was actually what was causing the cell competition. And so to do that, we decided to generate phosphorylated EF2 alpha independently of any of minute or of XRP1 function. And we did that by depleting the phosphatase, which is called PPP1R15. In clones of cells that deplete the phosphatase indeed have phosphorylated EF2 alpha, and they disappear from wing imaginal discs. So from this experiment, we could very easily have concluded that EF2 alpha phosphorylation was the mechanism whereby XRP1 leads to cell competition, had we not done one more experiment. So to be sure that EF2 alpha phosphorylation was downstream of XRP1, we just checked that XRP1 was not expressed in these cells. And contrary to our expectation, we found that phosphorylating EF2 alpha actually leads to robust induction of XRP1 expression. And that XRP1 is actually required to eliminate the phosphatase knockdown cells. So in fact, these cells that have high EF2 alpha phosphorylation are eliminated because they induce XRP1 as well, and XRP1 is also controlling their elimination, presumably by some other mechanism. And accordingly, we can rescue RPL36 heterozygous cells by knocking down XRP1, but a mutation of PERK does not rescue. So what we think then is that XRP1 probably regulates cell competition through transcriptional targets. We've shown that XRP1, as a heterodimer with its binding partner IRBP18, is a sequence-specific transcription factor, and it controls the transcription of about 80% of the genes whose transcription is altered in minute wing discs. And it's perhaps interesting to note that it doesn't control only single copy genes, but it also controls some transposable elements. So for example, the retrotransposon copia, expression and transcription of copia transcripts are induced about 20-fold in minute wing discs in an XRP1 dependent fashion. And the regulatory region of the copia retrotransposon contains 8 binding motifs for XRP1. So we wanted to know what might be a mammalian equivalent of XRP1. So XRP1 is a very rapidly evolving gene. We're not able to identify mammalian counterpart by sequence similarity. But IRBP18, its heterodimer partner, is the Drosophila homologue of CEBP proteins. So we looked for mammalian BZIP proteins that heterodimerize with CEBP and might have similar properties. And we came upon this gene, DDIT3 CHOP. And we found that if you overexpress DDIT3 in flies, you get a similar phenotype to overexpressing XRP1. Now, DDIT3 CHOP is a mammalian protein that functions in the unfolded protein response. And it's particularly important for enabling the unfolded protein response to kill cells through apoptosis. And so the DDIT3, so that XRP1 looks rather like a gene that performs a similar function in Drosophila to that which DDIT3 CHOP performs in mammals. It's at a slightly different place in the pathway. So in mammals, DDIT3 CHOP is induced by ATF4, which is a downstream target of EF2 alpha, whereas in flies, XRP1 is induced by EF2 alpha directly. And then but otherwise it looks fairly similar. And then in flies, ribosomal protein mutants can activate XRP1, which then can also phosphorylate EF2 alpha. So in some ways, XRP1 looks like a fly equivalent of DDIT3 CHOP. So what I've shown you so far then is that XRP1 is really looking like a central regulator of cell competition that can be activated in several different ways. It can be activated by mutations in ribosomal protein genes, and also, though I haven't shown you this tonight, by mutations that affect ribosomal RNA synthesis, and that all goes through RPS12. It can be activated by phosphorylation of EF2 alpha and by some other factors that affect translation initiation, and indeed, labs like Don Rieu's lab and Eugenia Pedini's lab have also shown that EF2 alpha phosphorylation leads to XRP1 induction, and Igaki's lab in Kyoto has shown that unfolded proteins ER stress can actually lead to cell competition, again through induction of XRP1. And so it's quite possible that other conditions that lead to EF2 alpha phosphorylation, like for example, viral infection, could cause cell competition, but that's not demonstrated. And then I haven't shown you this today, but defects in translation elongation also activate XRP1 and cause cell competition. And we think actually that several of the other kinds of cell competition that have been described by other people probably also act through XRP1. And then XRP1 controls translation through PERK activity in EF2 alpha phosphorylation and competitiveness by another mechanism, possibly through other transcriptional targets. So I want to move on now to talk about functions of cell competition, and I'm going particularly to talk about its role in aneuploidy surveillance. So aneuploidy is the major cause of human birth defects and miscarriage, and more than 90% of tumors are aneuploid. And in addition to the safeguards that prevent aneuploidy from occurring, if aneuploid cells do occur, it turns out that there's quite a lot of evidence that they can be eliminated from tissues. So this is a figure from a paper showing that mouse embryos constituted of mixtures of aneuploid ES cells and normal ES cells give rise to 100% normal mouse pups because the aneuploid cells get eliminated. And about 10 years ago, at the fly meeting, I heard an inspirational talk from Laura McNamee, who was a graduate student in Michael Brodsky's lab. And their idea was that the fact that ribosomal protein genes, that aneuploid cells could be being eliminated by cell competition because they affect the copy number of ribosomal protein genes. So there are 80 ribosomal protein genes. They're spread throughout the genome. This is the human chromosome complement that I'm showing here. And so many aneuploid genotypes are going to affect ribosomal protein gene dose, and they could be eliminated by cell competition. We decided to test that model. And so my student, June, developed a system that exploited the vast collections of FRT-containing elements generated by XLXsys and by the Drosdell project. And if these FRT-containing elements are placed in cis, then flip recombination will excise a section of the chromosome and generate a segmental aneuploid cell. And if the configuration of these elements is appropriate, such that the mini white genes are excised at the same time, then the aneuploid cells can be recognized as white clones in the eye. And so June found that when eyeless flip is used to generate these excisions in all of the eye cells, then generally one obtains a fairly normal looking white eye. So many of these segmental aneuploid genotypes, even though they are heterozygous for deletion of hundreds of genes, they survive fairly normally in the eye. But when heat shock flip is used to induce sporadic clones of these genotypes, we found that they only survived when they did not include any ribosomal protein gene, and that deletions that did include ribosomal protein genes were lost. Yet, if the whole head is of these genotypes, then they survive. And we studied this over many chromosome regions and many ribosomal protein genes, and came to the conclusion that cell competition due to ribosomal protein gene dose is a major mechanism for eliminating aneuploid cells. So we've generated RPS12 mutant mice, and we are hoping to find out whether they accumulate aneuploid cells. But there is already another genotype in mammals that in which cell competition is expected to be defective, and that's mutations in ribosomal proteins themselves. So we know in Drosophila that when the whole animal is minute, cell competition no longer occurs, and that is because there's no longer any, so if, for example, an aneuploid cell is generated, there's no longer any difference between the aneuploid cell and its neighbors because all the cells already lack a ribosomal protein gene. And so aneuploid genotypes that cannot survive in sporadic clones in the eye can survive if the animal is mutant for another ribosomal protein gene. So in humans, mutations in ribosomal protein genes lead to a disease called diamond blackfan anemia. And one feature of diamond blackfan anemia is a 5-fold elevated lifetime cancer risk. Now, we don't know if that cancer risk is due to inability to a failure of cell competition in those individuals, but it's one possibility that it is. Another difference between humans and Drosophila is the role of P53 in cell competition. So in many of the examples of cell competition that have been described in mammals, cell competition depends on P53. But P53 is dispensable for cell competition in Drosophila. However, XRP1 is a P53 dependent gene. Its name stands for X-ray induced P53 dependent number 1. It's actually the major P53 target in Drosophila. It just wasn't worked on because the mutants had no phenotype. And so indeed, XRP1 expression is induced in imaginal discs by radiation in a P53 dependent manner. And in fact, many of the genes that we normally think of as P53 targets are actually XRP1 targets. So HIT GFP, for example, is an XRP1 target. So what we think is that in mammals, P53 is often involved in cell competition. For example, in mammals, ribosomal protein mutations activate P53. But in Drosophila, they bypass P53 and activate XRP1. So we think that XRP1 in Drosophila is perhaps fulfilling some of the functions that mammalian P53 fulfills. So if that's the case, then if we create cells in Drosophila that have different P53 activities, we ought to see competition between them. And so my postdoc, Chaitali Khan, set out to test this. She made clones of P53 mutant cells in wing discs, compared them to their twin spot controls, and as is well known, they have no effect on growth. But then she generated a mild genotoxic stress by feeding the larvae a low level of cisplatin. And what happens then is dramatically different. The P53 mutant clones grow very big and their twin spots grow very small, showing that as expected, cells with different levels of P53 will compete in Drosophila as they do in mammals. It's just that in Drosophila, many of the genes that we see causing cell competition seem to act on XRP1, which is the gene downstream of P53, whereas in mammals, it seems like P53 activity itself that is targeted. Of course, this raises the question of how much of the tumor suppressive function of P53 is mediated by cell competition, and that's something else that we don't know. So today, I've shown you that cell competition is connected to several kinds of stress response pathways. I've shown you the connections between the ER stress response and the DNA damage response. In the ER stress response, XRP1 seems to be playing a role that's somewhat similar to a mammalian transcription factor, DDIT3 CHOP. In the DNA damage response, XRP1 seems to have acquired some functions that in mammals are mediated by P53. Now, both of these stress response pathways share a property, and that is that they control downstream effectors that can either repair damage to cells or kill cells. And it's generally thought that the stress response pathways decide between these two fates according to what would be most adaptive for the organism. But if you think about it, you can see that these systems can't be optimized unless the status of the neighboring cells is taken into account. And that's because you can imagine a cell, like this cell here, that's received some damage, and it's actually one of the least damaged cells in the tissue and therefore should be preserved. But in another context, a cell that has received the identical amount of damage could be one of the most damaged cells in the tissue, and it would be adaptive to eliminate it. So truly optimizing this system can only occur if the status of the neighboring cells that would be the potential replacements are taken into account. And this is how we're beginning to think about cell competition as a process that biases the decision of stress pathways between repair and replacement according to the genotype of neighboring cells. And we think that that's what you're seeing in a picture like this. And it's interesting in this regard, perhaps, to think about the effects of ribosomal protein mutations in different organisms. So in yeast, which has no P53 and no apoptosis, and where it's every cell for itself, in yeast, depleting ribosomal proteins causes a stress response, which is actually protective. So ribosomal protein depletion in yeast increases longevity, prolongs lifespan. So in Drosophila and in mammals, ribosomal protein mutations do not prolong lifespan. Now in C. elegans, which is also a multicellular organism, but where because of its strict lineage, hardly any cells can be replaced, in C. elegans, ribosomal protein mutations also prolong lifespan. So it's possible that the deleterious effects of minute mutations in flies and of ribosomal protein mutations in mammals are perhaps collateral damage of pathways that are designed to help eliminate sporadic damaged cells, and which unfortunately lead to detrimental effects when the entire organism is affected. So I'd like to thank our collaborators, people who sent us stocks, our funding. Today I've told you quite a few things. Cell competition replaces cells in intact tissues. Haploinsufficiency of ribosomal protein genes behaves like gain of function of RPS12. RPS12 turns on XRP1, which is a major regulator of the minute phenotype and of cell competition. XRP1 controls translation through reactive alpha phosphorylation. One function of cell competition, at least, is to eliminate aneuploid cells. And XRP1 behaves as though it has functions of the mammalian DDIT3 CHOP and P53 proteins, and the differential P53 activities lead to cell competition in Drosophila as they do in mammals, and that maybe cell competition allows organisms that have regulative development to optimize repair and replacement strategies. This is my last lab photo taken before the pandemic. And many of these people have left the lab now, and a few new ones have joined, but we are still intensively recruiting if anybody would like to join us, and I'll take questions. Thank you.
We have time for one question. So is it Pedro or Don who's going to ask the question?
My question is very quick. So what is the connection between the minute and PERK activation?
Yeah, what's the connection between minutes and PERK activation? We're not completely sure. There is a transcriptional stimulation of PERK, XRP1 dependent transcriptional stimulation of PERK. And there is also XRP1 dependent repression of other genes that reduce ER stress. And so it's conceivable that those transcriptional changes are responsible for PERK activation in minutes, but we are not sure if that's the explanation.
And there's a couple of questions from the chat, but I'm just going to ask you one of them, Nick. Do minute flies accumulate aneuploid cells in their bodies?
Do minute flies accumulate aneuploid cells in their bodies? Yeah, that's a good question. And I don't know of any data that addresses that. We haven't done anything that would address that.
Okay, let's give Nick a big round of applause.
Let someone else deal with that.
Okay. Just waiting for your slides. Is it starting? Okay, our next speaker is Heather Broyer from Case Western. I'm so sorry. Wasn't that supposed to go? There we go. And
Great.
I'm just gonna put this back. Okay. Yes, I'll give it back.
As opposed to Nick, my timer's already started for me, so we're working on that. I'd really like to thank the organizers for this huge honor. I'm really so happy to be able to be here and talk to you about our work today. So Theseus is a mythic king of Athens, and he went on many adventures in a boat that might have looked something like this, where he battled a minotaur and fell in love with Ariadne and returned in his boat to Athens, where the Athenians were so impressed with him that they memorialized this ship in a museum, kind of an outdoor museum, and meant to keep it in like seaworthy shape. And so over hundreds of years, they replaced it oar by oar and kind of plank by plank until the entire ship had been replaced. And philosophers, beginning with Plutarch in the 1st century, posed the kind of paradox of the ship of Theseus. If the entire ship has been replaced, is it still the same ship? So, and philosophers think about that in terms of identity. And so we can kind of transpose that question from thinking about personal identity to thinking about cellular identity and ask, how is it that cells are able to balance the kind of rigid aspects of cell fate with the kind of flexibility that they need in order to change with changing environment? And that's what I'm going to talk to you about today. So classic, pioneering, really elegant, influential studies, largely from Oliver Hobart's lab and Christot's lab, have led to models of combinatorial coding for neuronal identity. And so in these models, it's proposed that it is the precise complement of overlapping transcription factors across all of the hundreds of neuronal cell types that leads to distinct identities. And it's been shown that the effectors of these transcriptional codes are really critical in establishing core features of neuronal identity, which include axon guidance, target matching, and neurotransmitter identity. But in addition to these fixed aspects of neuronal identity, it's totally critical that neurons are able to modulate their behavior to adapt to a changing environment. And it's these kind of plastic features of neuronal identity that are really essential for a lot of the kind of higher order functions of the nervous system, like learning and memory, that are kind of the extraordinary aspects of neuroscience that we kind of want to understand. So what I'm going to talk to you today is work from our lab that I think argues that neurons and glia have to balance transcriptional codes that hardwire aspects of neuronal identity, which those enable flexibility to enable neurons to adapt. I'm going to first talk about our work looking at neurons that speaks to this, first in axons and then in dendrites, and then I'll talk to you about sulfate plasticity in glia. So what does a neuroscientist mean when she says structural plasticity? Totally simple. She just means that the neuron changes its shape over time in response to stimulus or environment or condition. So here, so synapse, presynaptic side, postsynaptic side, the presynaptic side releases neurotransmitter and the postsynaptic side responds. So structural plasticity is very well understood, both in presynaptic and postsynaptic compartments. So here I've drawn a postsynaptic compartment. So in the brain, excitatory postsynaptic compartments are housed within dendritic spines, and in response to increased activity of the synapse, spines both grow in size and increase in number to strengthen neurotransmission. Similar thing is seen on the presynaptic side. So presynaptic neurotransmitter is released from active zones, and active zones are housed within boutons, which are just presynaptic varicosities. And if you drive the neuron, if you stimulate it more, the neuron responds by budding more boutons. We're Drosophilists. We study this at the Drosophila neuromuscular junction. So this is the beautiful neuromuscular junction prep that you saw in Christo's keynote address. In the middle there, you see, so the developmental unit of this system is the hemisegment. Those are motor axons that are coming out from within one segment and innervating in a totally stereotyped way, muscles in the periphery. On the right, that beautiful little guy, that's the neuromuscular junction that forms on muscle 4. And so what I want you to take from this is, under normal conditions, this is beautifully stereotyped. So at muscle 4, normal conditions, there are 20 boutons, like plus or minus 2. So it's totally hardwired. But if you push the system, if you drive the motor neurons harder, either you can do it optogenetically, you can do it with electrical stimulation, you can do it with spaced high potassium, the presynaptic terminals respond and they grow. Okay, so you're budding new boutons in response to increased activity of the motor neuron. How do we know they're new? We know they're new because they have only a presynaptic marker. They haven't had time to put down the postsynaptic marker. So in this slide, you can see them as HRP positive and DLG negative. This is just to show you that these aren't weird little things that don't differentiate. If you give them time, they'll turn into self-respecting boutons with real synapses in them. So here they are. This is work from Vivian Budnick's lab. They're laying down both presynaptic and postsynaptic markers over time. So these are bona fide boutons. How did I get interested in this? Well, I've been obsessed with the transcription factor FOXO since I was a postdoc, and I've been obsessed with it because of that beautiful expression pattern in the ventral nerve cord. So when you if you look at FOXO in the ventral nerve cord, it's expressed in a highly stereotyped group of neurons that really overlaps the motor neuron population that's co-labeled there with PMAD. One would think, and I certainly thought as a new assistant professor, that given this beautiful expression pattern, the phenotype would be super obvious. And it totally was not super obvious. And it took a really persistent graduate student in my lab named Ina Nechaparenko to find a FOXO loss of function phenotype. And so what she's shown is there on the right. When you lose FOXO, specifically in motor neurons, what you see is an elevated distribution of stable microtubules at presynaptic terminals. And I'll show you in detail what that phenotype looks like later, but just remember that there's too many stable microtubules. So we wanted to ask whether this was required for plasticity. And this was interesting to us because microtubules you talk to neuroscientists, what neuroscientists are interested in synapses is actin. What microtubules are thought to do is provide the stability to the neuron. They enable it to, adapt its beautiful form, and they are responsible for motor movement to terminals, but actin does the interesting thing at synapses. Because we had this microtubule phenotype, we wanted to ask whether microtubules were required for presynaptic structural plasticity. I'll say that microtubules, super broadly speaking, display two different types of plasticity, two different types of dynamics. On the left is polymerization and depolymerization of a single microtubule. Okay, so this is just catastrophe and rescue of one individual tube. On the right is a less understood form of plasticity called sliding, and this is kind of network dynamics. This is tubes sliding against each other. And that movement of microtubules on microtubules is driven by kinesin motors. So do dynamic microtubules promote structural plasticity? So we ran the assays both in wild type and in FOXO null mutants. And here we're looking at presynaptic in green and postsynaptic in magenta. And what you can see in wild type is there's a twofold increase in these nascent boutons when you stimulate, and that increase is lost in FOXO mutants. So that says that FOXO is necessary for activity-dependent presynaptic structural plasticity. This was enough for us to go ahead and try to figure out the pathway it was acting on. And so I'm going to walk you through three things we looked at: identify the upstream factors, look at the downstream factors, and try to understand what it's actually doing. So what's regulating FOXO nuclear localization? There's a beautiful expression pattern. We took kind of a brute force approach and just screened for receptors that had been implicated in FOXO-nuclear shuttling and other systems. None of the kind of most likely candidates were involved. We remembered TOL6. This is a TLR that in flies is nicely expressed in the CNS, which is what I'm showing there. So a really outstanding graduate student in my lab named Colleen McLaughlin just looked at TOL6 mutants to ask if nuclear FOXO was affected, and it is. So we see about a 50% decrease in FOXO-nuclear localization in TOL6 nulls. Okay, so once a TLR is involved, then you want to look at the adapter protein that might be downstream. TLRs signal through TEER adapter proteins. There are only two in flies, MyD88 and DSARM. Wasn't MyD88, but DSARM, loss of DSARM gives an equivalent phenotype to loss of TOL6. Both of them block FOXO-nuclear localization. This is our proposed pathway. I just want to walk you through a teeny bit of epistasis that we did so you believe what I'm going to tell you for the rest of the talk. Again, so here we're looking at NMJ4, which is the NMJ that I pointed out at the beginning of the talk, 20 boutons in controls. You see an increase when you overexpress FOXO. Also see an increase when you overexpress TOL6. That increase is lost in a FOXO mutant background. So that says that FOXO is downstream of TOL6. Like I said, TOLs signal through TEER domains. We wanted to ask if the TEER domain was required because there's evidence from olfactory system that TOLs can act in targeting in that system independent of their cytoplasmic domain. If you overexpress TOL6s that lack the cytoplasmic domain, it doesn't lead to overgrowth, so you need that TEER domain on the cytoplasmic side. Overexpression of DSARM phenocopies, overexpression of TOL6 or FOXO. That overexpression phenotype depends on FOXO, so FOXO is downstream of SARM, and overexpression of TOL6 depends on SARM. So if you followed all that, we did the genetics, and it really supports the idea that TOL6 is upstream, running through DSARM, and FOXO is downstream. Okay, and this is what the phenotypes actually look like. So we're now looking at stable microtubules labeled with MAP1B in presynaptic boutons. Okay, and here MAP1B is called FUTCH, labels stable microtubules. We and others have found that NMJs that are characterized by increased microtubule stability have too many microtubule loops. And that's really thought to reflect kind of increased looping instead of depolymerization. And what you can see in all of these null backgrounds, we see an elevated number of microtubule loops, consistent with the idea that FOXO signaling promotes dynamic microtubules at the NMJ. Okay, cool. I hope I've convinced you that these proteins regulate FOXO nuclear localization and somehow dynamic microtubules. What are they running through? So we did a small QRT-PCR screen trying to identify genes that might be regulated by FOXO. And we really thought we were doing this on the way to a larger RNA-seq kind of experiment, but we got lucky. So we picked about 50 microtubule and actin regulators, and one of them instantly jumped out to us. It's a mitotic kinesin called Pavarotti, and it's called Pavarotti because cells don't divide right and they get too big in embryos. So it's a great Drosophila name. And we saw about a 5-fold increase in Pavarotti RNA levels in TOL6 mutants. Okay, we backed this up with an antibody. There's Pavarotti at presynaptic terminals, and you see increased Pavarotti about 2-fold in TOL6 mutants and FOXO mutants. So what these data say are that FOXO signaling normally represses Pavarotti expression. When we actually looked at the literature, it made pretty much sense. So Pavarotti, the mitotic kinesin Pavarotti has been shown to inhibit kinesin 1 dependent microtubule sliding, and work from the Gelfand lab has argued that this actually is required in neurons where Pavarotti is required to inhibit the movement of microtubules against each other during axon outgrowth. Okay, so if that's the case, if we're onto something here, then the increased microtubule stability that we see in these mutant backgrounds would be driven by, caused by elevated Pavarotti expression. If that's the case, then the phenotype of Pavarotti overexpression in NMJs should look like the loss of function phenotypes that we saw in TOL6, FOXO, and DSARM. And I wouldn't be here if that weren't the case. So if you overexpress Pavarotti exclusively in motor neurons, you get the loss of function phenotypes of FOXO pathway out. So that says that Pavarotti clip is sufficient, the Pavarotti kinesin is sufficient to stabilize microtubules at the NMJ. Okay, so we thought this was running through Pavarotti. we really wanted to look more closely at microtubule dynamics in presynaptic structural plasticity. This is the assay that I showed you before. FOXO, TOL6, and DSAR mutants all block the ability of NMJs to grow new boutons with spaced hypotassium stimulation. That's the data. This is the data shown here quantified. The left is the nulls and the right is motor neuron specific knockdown. So TOL6 FOXO signaling is required specifically in motor neurons for activity dependent structural plasticity. Okay, does Pavarotti have anything to do with this? We reasoned that if elevated Pavarotti expression in these backgrounds was responsible for the failure of presynaptic plasticity, then you might expect that this phenotype would be suppressed by decreasing the dosage of Pavarotti. So if this is caused by elevated Pavarotti, then if we decrease the dose, we might suppress, and that's the case. So you can see that all of these phenotypes are strongly suppressed by loss of one copy of Pavarotti. Okay, so this is the model coming out of this work where we propose that this pathway establishes competence for presynaptic plasticity for maintaining a pool of dynamic microtubules at NMJs. Okay, we wanted to move from looking at axons to shift and looking at dendrites. And that's because, so axons are the signal sending component of the neuron and dendrites receive the signal. And axons have much more, in general, have much more stable microtubules than dendrites do. And so if we were onto something, then we expected to see a phenotype in dendrites. So that's what we set out to look at. So what we found is that FOXO is required in sensory neurons to establish proper microtubule dynamics. So here we're looking at these beautiful nociceptive sensory neurons that form a tile against the body wall in larvae. So these are huge elaborated neurons. And so here you're looking at controls on the left and FOXO on the right. And what you can see is that the FOXO mutant cells are much less branched than the controls. And actually what this particular experiment is sequential imaging of the same cell in vivo so that we could actually look at branch dynamics because we wanted to know, we knew they were less branched and we wanted to know why. And so maybe it's too small to see, but my students, so this was work done by a previous student in my lab, Jim Sears, and all of the new branches at 2 hours have a green arrow and all of the lost branches are marked with a magenta arrow. And what we could see is that in controls, there's more gain than loss, and in the mutants, it's the opposite. Okay, and so that led to over 2 hours, a pretty robust increase in branching in controls and not much of anything going on in FOXO mutants. So this says that overall, FOXO promotes the initiation and stabilization of new dendritic branches. We were like excited, but we had no evidence that it had anything to do with microtubules. What we did next was to look at our friend FUTCH. This is the microtubule stabilizing protein, to look at its distribution in dendrites. And if you just focus on the magenta panels, what I hope you can see is that in controls, FUTCH labels that primary branch really well, but not the secondary branches. If you look at the FOXO mutant, what I hope you can see is that the primary branch is lit up, as is the secondary branches. So that says that FUTCH the distribution of FUTCH is increased in FOXO mutants, which is consistent with the idea that FOXO normally limits the distribution of stable microtubules and dendrites, which was consistent with our hypothesis. Okay, so the last experiment that I'm going to talk to you about dendrites was actually super exciting to us. So the organization of microtubules and axons and dendrites is different. In axons, they're plus end out, and in mature dendrites, they're mostly minus end out. Okay, but when dendrites are growing, when they're young and vibrant, more of them are plus end out. Okay, so we were curious to see whether FOXO, overexpression of FOXO would increase the population of plus end out microtubules and dendrites. And we did this by using EB1, which is a plus end binding protein, and doing live imaging. Okay, so these are kymographs. You're looking at movement of EB1 over time. And what you can see, so you're looking at two different stages of third instar here, kind of early and middle, when the microtubules are really dynamic. And so most of the EB1 comets are moving retrograde. The ones with the purple arrows, those are the anterograde comets. Okay, and you can see that in controls, there aren't very many at all. But when we overexpress FOXO, we have a really robust increase in the number of anterograde microtubule comets. So this says that FOXO is sufficient to drive anterograde microtubule polymerization, which was exciting to us because this is a characteristic of young growing dendrites. Does the animal care? I told you these are nociceptors, they sense pain. So if the animal cared about this, you would expect them to have decreased pain response, and they do, which is what I'm showing here. Across the top, you're seeing that they move fine. Those are just traces of the larvae crawling around on a plate. There's nothing wrong with their motility, but they don't respond appropriately to a 50 millinewton stimulus. So FOXO is required for normal nociceptive response. So together, what our data say is that neuronal FOXOs protect morphological plasticity, both in the axonal compartment and in the dendritic compartment. And I'll just say that this feature of FOXO is really well conserved in mice and worms. So evidence from Azadbani's lab and Daniel Cohen-Ramos's lab really support the idea that neuronal FOXOs are required for morphological plasticity. Okay, just for the last couple minutes, I'm going to talk to you about glia. And this is because, so this is data from the Eric's lab looking at, this is a t-SNE plot looking at single cell data from adults. Okay, so neurons, the big kind of island in the middle, those are all different neuronal populations. Glia are at the top, and you can tell that because Repo is a pan-glial marker. Okay, so this is adult single cell data. What you can see in the adult FOXO is almost entirely a glial gene. Okay, it's co-expressed with Repo. Totally, like, I felt very validated, but really happily, DSARM is also really a glial gene. So what are these genes? Why are they so highly expressed in glia? So if you know anything about glia, we refer to them as this kind of support cells of the nervous system. And unlike neurons, which hyperspecialize, glia really retain multipotency over life. They're really context-specific supporters of neurons. When a neuron is sick, glia might phagocytose it. In other situations, it might provide trophic support. So what they do completely depends on the needs of the neuron around them. So they, in some sense, display more cell-fate plasticity than the hyperspecialized neurons do. I'm going to talk a little bit about phagocytosis. So I just want to tell you that a ton of neurons die in the developing nervous system, and glia are responsible for taking care of the mess. So they are responsible for clearing neuronal corpses, for clearing neurites, and also clearing synapses. All of that is driven by glial phagocytosis. We're specifically going to talk about cortex glia for a few minutes. So these are cell body associated glia in the nervous system that provide work from Jada Coutinho-Budd shows that they provide trophic support, but they also are responsible for phagocytosis. Why were we interested in this? We initially fell into this because TOL6 mutants were published as having increased apoptotic debris in the brain. And so we had these new components of a TOL6 pathway, and we wanted to ask whether they also displayed this phenotype. These are the nulls for the pathway. You can see, so, and this is labeled with DCP1, which is a activated caspase that labels apoptotic cells. We could see in the mutants, in the single mutants, as well as in the TOL6 FOXO double, really pronounced increase in apoptotic debris in the CNS. This is showing that it's neuronal debris because we can block it with overexpression of P35, which is an apoptosis inhibitor specifically in neurons. We assumed it was neuronal. My student did the experiment, knocking down pathway function in neurons and got no phenotype at all. I might have been slightly skeptical. She repeated it using a glial driver, and we got the phenotype, the null phenotype, right back out. So this is a glial function of this pathway. What type of glia? Beautiful drivers that Mark Freeman developed for all different flavors of glia. The phenotypes here were pretty clear. Only when we knock down this pathway in cortex glia do we phenocopy the null phenotype. So that says that FOXO pathway functions in cortex glia to limit apoptotic debris. So, and I didn't show you data, but DSARM and FOXO also function in cortex glia to limit apoptotic debris. How? They regulate Draper. So Draper is the key engulfment receptor in glia that's responsible for phagocytosis. So it's like the prime suspect. And what we found in pathway mutants is that Draper expression in cortex glia, you can see this across the middle, the cortex glia are highlighted in green. Draper levels are about 50% reduced. So that says that this pathway is required in cortex glia to sustain Draper expression. Draper is actually the critical regulator of this phenotype, because if we drive Draper specifically in cortex glia in these backgrounds, we perfectly suppress the phenotype. Okay, so I'm skipping the rest of this story. What we could also show was that Schwetzler 5, which is a TOL6 ligand, is released from cortex glia, released from neurons, excuse me, binds TOL6 on cortex glia to activate this pathway and prime glia for phagocytosis. I'm really obsessed with FOXO, and I wanted to understand how in the world DSARM linked to FOXO. So we went after, FOXO is really a signal integrator and can be regulated by many different kinases. So we went after the kinase. Work in motor neurons, he suggested to us that it was likely a MAPK cascade. So we screened MAP3Ks in the fly and landed on a MAP3 kinase called ASP1, okay, which turned out to be actually a really good, the literature fit. It's widely implicated in RS-mediated signaling and has been shown to be required in other contexts for ROS-dependent activation of FOXO. So on the left, you're looking at our DC, this is a MAP3K. On the left, you're looking at cortex glia knockdown of SARM and then ASP1, and you can see that they perfectly phenocopy. There wasn't an ASP1 null, so we made one, and we see the same phenotype in ASP1 nulls that we do with cortex glia knockdown, and it's required again for expression of Draper. So finally, the last thing I'm going to show you is that if you know anything about SARM, you actually, unfortunately for me, don't know anything about our work. What you know about SARM is that it's a critical executioner of axon degeneration. This is work from Mark Freeman and Aaron D'Antonio and Michael Coleman and Jeff Milbrand. And so what they found, really surprisingly, this looks like a normal run-of-the-mill pterodactyl protein, but actually it's an NAD hydrolase. And when it gets activated, when an axon is injured, it leads to catastrophic energetic loss and axon destruction. So we really wanted to know whether the NAD hydrolase activity of DSARM was required for its signaling function. So that was the first question. The second question was, for axon degeneration, SARM forms an octamer, okay? And that octamer is driven by SAM-SAM multimerization. And we wanted to know whether that SAM multimerization was required. We built alleles to test this. So we knocked in a series of domain mutants as well as an E to A mutant. And I'm going to show you the phenotypes on the next slide. So what we found was that knockout of SARM or the E to A mutant give the same increase in apoptotic debris. So that says that the NAD hydrolase activity of SARM is required for this pathway. Totally surprisingly, we found that the SAM domain, so we made a mutant that is just arm tier. When we look at DCP1 levels in that mutant background, completely wild type. So that says that the NAD hydrolase activity of DSARM is required for signaling, while the SAM domain is dispensable. Okay, so altogether, this is the pathway that I've shown you. Dying neurons signal through TOL6, through SARM, which is acting as an enzyme to somehow activate ASP1 and drive FOXO into the nucleus to prime glia for phagocytosis. Does it matter? It does. So if you knock this pathway out, specifically in cortex glia, we looked for degeneration because it's been proposed that elevated levels of apoptotic debris in the brain lead to neuronal degeneration. And we scored this in adults by counting vacuoles. If you knock this pathway out in adults, so if you look at the middle bars there, that's 10 days post-occlusion, you see an increased levels of vacuolization in mutant backgrounds. So that says that FOXO, this glial signaling pathway, normally protects the brain from early onset degeneration. You take one thing from this talk, I hope you, so FOXO functions are really well conserved between worms and flies and mice, okay? And in general, overall, what FOXOs do is promote neuronal youthfulness and glial youthfulness in order to enable these cells to remain nimble and respond to changes in condition. When I was making this slide, I was thinking that my lab itself bore some resemblance to the ship of Theseus as it changes over time and all of the components are replaced. And, you know, I think when really fantastic students leave the lab, it can feel really kind of crippling, and then we're replenished when wonderful new students join. So the people in my lab who've worked on the FOXO project, I listed in bold. And then I have actually four current students in my lab who are here today: Liz Anderson, Marina Bostelman, Dan Jindal, and Jarek Ridbaum. I'd like to thank our collaborators and also our funding. Thank you.
Hi, this is Unmila. I'm from Marquette University, Wisconsin. So did you look at the glial morphology in these phagocytosis?
Yeah.
So like if you do, like how do they look?
They look fine.
So there is no issues with their processes or anything?
No. Did we run them through every like high resolution test of morphology? No, but at a gross level, they look fine.
Okay, and the other question, if I may ask. So did you also look at the neurons that are kind of like close to those glial cells where you look? So they look fine as well?
We didn't see any changes in neuronal number in any of these mutant backgrounds. Again, we didn't do an exhaustive test of every neuronal subtype, but we didn't see any changes in number.
Okay, thank you. Good talk.
Hi. Oh, sorry, we have to move on.
Oh,
So please give Heather a round of applause.
Yes, our next speaker is Yanlan Mao from University College London, and she'll talk about coping with mechanical stress and tissue dynamics in homeostasis and repair.
Fine, let me just get rid of this. All right. Sorry, I'm a bit short. Let's lower this. Good evening, everyone. So thank you for staying awake and still being here tonight. So first of all, I'd like to thank the organizers for inviting me to this conference. It's a really good excuse for me to leave wet and cold London for the first time in 2 years, actually, and come here to sunny San Diego. So it's been really, really wonderful to see everyone here in 4D. And so, yeah, thanks for being here. And so in my lab, we're interested in how tissues get to the right size and shape and how this beautiful diversity that we see in nature is generated. And so, of course, we all know it's in our genes. After all, we look like our parents, our kids look like us. But exactly how do our genes actually give rise to the beautiful sizes and shapes that we see in nature? And you may have seen this little one running around the hotel recently in the last few days. Well, she's a bit older now. So of course, many of us have studied for decades, different omics approaches, looking at signaling networks, gene regulatory networks, protein interaction networks, and so on, to try to understand growth and morphogenesis. But we're still very far from really understanding, even if we know, the genetic details, how do we actually build and sculpt beautiful, tissue? And that's really because, growth and morphogenesis are very dynamic processes involving basically shape changes such as cell division, cell migrations, even cell growth. All of these are dynamic processes that require forces to act on object. So even biological objects have to obey the rules of physics. All right, so which is why in my lab, we really focus on the role of mechanical forces and how these forces actually shape tissues. And specifically, we're interested in 3 aspects. One is how do tissues actually get into shape during development? Another is how do they then stay in shape throughout the rest of the lifetime of the organism? And then 3 is how do these tissues get back into shape if they're accidentally injured, for example, during their lifetime? And so hopefully in today's talk, I'll give you, little stories from each of these aspects to give you an overview of the work we do in our lab. And first, I'll start off by looking at how tissues get into shape using an example of 3D tissue folding. And so I'm sure many of you know, just like this origami crane, many of our organs are highly folded, and the position of these folds are actually really important for the function of these organs. And so, by the way, this origami is my only data contribution to this talk. Sorry about that. But I think it's pretty cool, right? Anyway, and so I think a few talks earlier today mentioned that many labs, and actually amazingly many fly labs, have looked at the molecular and mechanical mechanisms regulating how to actually generate a fold or invagination. But many of us have not really looked at, how are these positions of folds generated? So we know how to make a fold, But how does the tissue know where to actually generate these folds in order to then buckle and undergo complex shape changes? And that's something we've been looking at in the last few years. So how does a tissue know where to initiate a fold? And so we use obviously Drosophila and the beautiful wing disc to look at this problem. So many of you, I'm sure, know the wing as a monolayer epithelium, reasonably flat structure, but actually in cross-section, you'll see that, if I can find my mouse, there we go. So in cross-section, actually the wing disc along the major axis has these 3 very precise folds. And these folds always form in exactly the same places at the right stages in time. So it's an incredibly robust system to look at folding morphogenesis. And so our hypothesis at the time was really that differential planar growth rates, so growth in the plane of this flat epithelium, actually induces folds to occur in the right positions. And the reason for this hypothesis actually stems from work during my postdoc, where I noticed that the growth rates in the wing disc are actually differential. So as you can see in this simulation, computer simulation, you'll see that the cells in the center are actually growing and dividing faster than the ones in the periphery. And as a result, hopefully you appreciate these peripheral cells are actually being stretched circumferentially, right? But this is only in 2D. The simulation is fixed, so it cannot come out of the screen into your faces, right? But in fact, in reality, in 3D, if these stresses could dissipate in 3D, you probably get this occurring. So I encourage you all to, stretch your shirts or your t-shirts. And we're not on Zoom, so actually I can see if you're doing it or not. So but I assure you this will happen. And so this is really where we decided to kind of test our hypothesis. And so importantly, I wanted to test it using computational modeling driven approach, which is a really important approach that we use in our lab. And I just want to emphasize this, and at least for me, a model is really not about trying to recapitulate everything that we see exactly as it occurs. If I could do that, then there's almost no point in doing the model. I already understand everything. But it's really about generating a minimum sufficient set of rules that then allows us to test our hypotheses and hopefully make predictions that we can then test in vivo again. And there's this constant iterative process between modeling and in vivo experimentation that really can drive a project forwards. And so just to emphasize this famous saying from a former colleague of mine at UCL. And so where do we go? So when this project started, a very talented postdoc of mine, Melda, worked in collaboration with Jose Munoz at UPC in Barcelona to generate a new computational model of a growing epithelium. So this work is published and the code is all online. I'm very happy to share it. It's all on GitHub. So I won't go into the mathematical detail today, but essentially what we did is to generate a model where we basically discretize the tissue into a set of prisms. And each prism, as you can see here, will have different mechanical properties and they can have different growth rates. And as a result, as the tissue grows and develops, stresses will build up depending on these different properties, and you'll get a tissue that's hopefully shaping into a complex structure. And so we started the simulation with starting parameters that we measure in vivo. In this case, it's a wing disc that doesn't have any folds yet. And so this is essentially what it looks like in the simulation. And so these different layers of elements just allows us to have different polarities. For example, apically, basally, the cells can have different properties, but it is a monolayer epithelium still, okay? And so then we ask, okay, what do we need to add to the model? What are the minimum sets of requirements to allow folds to occur in the right positions? And so just a sanity check, if everything is uniform, so uniform growth, uniform physical properties, there's no mechanical kind of resistance, then we basically see what we predict, which is a flat tissue growing pretty much infinitely flatly. So that's great sanity check. However, there are some very basic requirements that you need to add to the model to allow any folds to occur in the first place. So for example, you need to have some external resistance, providing compressive forces for the tissue to buckle. But also, in fact, for it to start to fold asymmetrically, the apical surface actually needs to be stiffer than the rest of the tissue. And this we tested. And so just to show you an example of this simulation here, you'll see that it's starting to look a bit like a wing disc, but the folds are still not in the right places. So what else do we need to add into the model? And so they remember the hypothesis is about the differential growth rates, right? And so Maria, another postdoc in the lab, really spent a lot of effort, all summarized into one slide, unfortunately, to really measure the detailed growth maps of the wing disc. And essentially what we did is to do clonal analysis, induce sparse single cell clones, let them grow for 24 hours over different 24-hour periods, count the number of cells in these clones, and there you get the growth rates. And we map this in detail, spatially and temporally. And then when we input this growth rate into the wing disc simulation, we start to get these 3 folds forming in precisely same positions as we see in vivo, which is beautiful. But again, anyone who's done any computation modeling can tell me that, I could probably create an elephant or a mouse on the screen if I wanted to, given the right parameters. And that is probably true. And so therefore what we had to do, and importantly for every simulation kind of conclusion, is you really need to keep all parameters same, but change just one single factor, in this case, growth rate patterns, to see what happens to that effect. And when we do that, by just making the growth now uniform, distributed throughout the tissue, what we now get is these uniform ripples throughout, rather than these 3 folds in the right positions that we see in vivo. So I think this is a pretty good test of that. But still, not good enough. As I said at the beginning, what we really need for models to do is to make predictions, right? Can we make predictions that we can then test in vivo? And so we did many predictions, but the one that's really, really interesting is we decided, okay, what if we test the model by making in silico mutants? So we mutated this critical region here, where we know the folds will form in what we call the hinge region. This is a fly audience, so you know what I'm talking about. And so we reduced this hinge region here to 50% growth. And what the simulation then predicts is that, in fact, now we will only get 2 folds rather than the 3 folds that we saw in vivo. And so this is a really simple, neat prediction that we can test in vivo, right? And so then it's where the power of fly genetics really comes in. I think this audience will really appreciate this work, because this got me really excited. So we set out to therefore find a genetic basic condition that we can mimic this phenotype with. And looking through lots of reasonably old literature, we found this mutant lying. It's a specific wingless spade enhancer, where it was sort of noted in the paper at the time that the hinge region specifically has reduced growth. And they saw the adult phenotype here, the hinge is smaller, but they didn't look at wing disc data and didn't really quantify the growth in much detail. And so this seemed like a promising avenue. So I took these flies and we confirmed that indeed growth just in the hinge region is reduced, whereas everywhere else is the same, which is exactly what we wanted to find. And amazingly, when my student Natalie then looked at these wing discs, she saw only 2 folds. So honestly, I had to, I almost cried when I saw this beautiful data. It was just so beautiful. And I had to count it several times, because it's like, is it 2? Is it 3? But it really is just 2 compared to the normal 3. And so to me, this really completes the circle of this kind of iterative loop between modeling and experimental work. And I think in conclusion, what we can say is that differential growth patterns in the plane of a tissue is really important, at least in initiating where folds should form. But of course, if you then have to form the fully looped complex structures, you need many other forces, such as apical constriction, lateral constriction, basal expansion, that many other labs have shown. And these extra kind of forces will really be required to stabilize these folds. But in terms of initiating them, these growth patterns could be really key in defining where they form in the first place. And so that's, really great. But as I said, you've taken so much effort to get into the right shape, but how do you stay in shape for the rest of your lifetime? And I think during the last 2 years of lockdown, we probably all had, challenges of trying to stay in shape. So, despite, our environments constantly changing, especially mechanical forces constantly changing around us, we really have to buffer these forces to stay in shape. And how do we actually do that, right? And so again, my lab has been looking at this aspect for a while. And one of the tools we had to generate to really ask very directly, how do tissues cope with external forces, is to come up with a device where we can apply a controlled physical force, a pure force application. So not through genetic means or pleiotropic effects, but really, apply a directed force and see how the tissue copes with this. So we designed this stretcher where, in this case, we can put the wing disc in between 2 layers of PDMS, and then apply this, basically a PDMS sandwich with a wing disc in the middle to a metal device, where we can then rotate the device left or right, and this will stretch the wing disc or compress the wing disc. And this we can now actually have on a motor. So we can do this in a cyclic fashion and so on. And so then we can image this live while it sits on the confocal core and observe how does the tissue behave in response to these mechanical stretching forces. And so one of the many things we looked at was to look at obviously how the ectomyosin, the mechanical machinery which defines the tissue itself, actually responds to the stress. So to cut long story short, essentially what we found is that within minutes of mechanical stretch, so in this picture here, you'll see the wing disc being stretched, whereas this is the control just being anchored in the tissue, in the stretcher. And within minutes, the ectomyosin structure polarizes to polarize basically along this line of stretch, whereas ectoterium, which is the control here, remains unchanged. Okay, and so we did a lot of work to then find out, in fact, the molecular pathway responsible for this is via diaphanous actin formant. And this really is responsible for polarizing this ectomyosin structure in response to forces. And so essentially what the tissue does is when it senses a stretching force, whether it's from neighboring cells or from, in this case, an exogenously applied force, it will mechanically remodel itself, change its material properties, and essentially stiffens up to resist these pulling forces, preventing it from deforming. And we actually show that wing discs that are mutants for diaphanous actually have a more deformed shape. And so this, I think is sort of our way to start to look at how tissues cope with these mechanical forces. But the stretch we have here only allows us to stretch the tissue ex vivo, and we can only do that for a limited time. So really maximum up to 5 hours. And then you really can't see what happens to the wing disc after this, right? We can't let it develop again back in the fly. And so what we've been doing recently is to develop some kind of in vivo stretching assay. And so this is very new, very preliminary work that I wanted to share with this audience because I think it's really exciting. And so what we've been doing is to design these PDMS stretchers, PDMS channels, where the larvae can crawl through these channels, but then they'll face these very constricted spaces where they have to really squeeze through these channels. And when they squeeze through these channels, this essentially causes the tissues inside to get deformed and stretched. So what you'll see here is through these movies. So we also have control channels where the larvae are happily crawling along. And you'll see in this movie that even during normal crawling, the labeled wing discs here are being stretched and compressed randomly as the larvae moves. But when they crawl through these constricted channels, you'll see that the wing discs actually have to undergo much more deformations. And especially you'll see in this one where the channel is so small that you see this really stretched wing disc just about crawling through. So this is the whole larvae crawling through, by the way, but only the wing discs are labeled so you can see them. And so hopefully you saw that, the last one really had to be so deformed in order to fit through these channels. And so this is a really great way to stretch these wing discs in vivo. And then we can let these larvae develop and see the consequences of this in vivo stretching. And I think we were amazed that the disc could stretch so much because this is way beyond the stretch we ever apply ex vivo. And so this I find really, really exciting because what we've now done is to scale this up. We can now have hundreds, tens of channels, and these larvae are crawling, in between these channels. And it's pretty amazing, they voluntarily crawl through the channels, even if there's no food on the other side. They just kind of get through. Yeah, no, we were really surprised. They really, really want to do it. And so what we can then do is to track individual larvae crossing events. And so initially we thought we could probably just get away with averages, but in fact, every larvae behaves differently. So you really need a one-to-one correspondence between larvae and the eventual phenotype. And so we do individual larvae tracking. And so this is an example of a track for one larvae. So you can see that at the beginning, it's really happy across every time there's a channel it crosses, but then it eventually gets a bit smarter and it kind of gives up. So it stays within one channel, but then it'll cross again and stays within that channel. And so then we can, give each larvae a specific identity and put them into these 96 well plates and let them hatch in these larvae. Eventually they'll turn into adults and we can look at whatever phenotype we want to look at, including survivability, wing size, wing shape, and so on. And so as I said, this is really early work. There's very little data, but already have some to show that it does seem like with the channels getting tighter and tighter, the flies do survive less and less. So there's clearly an effect on the flies using these stretching devices. But of course, we are still early days in analyzing, for example, what does that do to the wing shape? What does it do to the wings patterning, as well as many other features we'd like to look at. So we're very excited by this work. So watch this space, and it'll be really nice to get some feedback from people here. And we're very happy to share this device if you want to stretch, whatever organ you're interested in the fly larvae. So this could be
of course, we can scale the channels to whatever dimensions you want to look at. So it doesn't have to be flies either. So more to come there. And so finally, moving on to the last part of the talk, which is to ask, so these stretching forces obviously are really great, but what if they become so extreme that they actually damage the tissue, right? And so the salamander is brilliant at regenerating its limbs and again every time we chop it off, but most of us are not very good at that. So how can we improve our mechanisms of tissue repair to really cope with injury during our lifetimes? And so the wing disc, again, as many of you know, is a brilliant model to look at tissue repair. And so what Rob, another brilliant postdoc in the lab, has done is to develop these wounding assays where he uses a 2-photon laser to ablate a patch of cells and then record the cellular dynamics as the tissue repairs itself after the injury. And so what he saw specifically is that during the repair process, there are these intercalation events at the wound edge, where basically, as the tissue closes, the wound edge cells one by one leave the wound edge such that the tissue, the hole can close seamlessly. And so as you can see here, this dark blue cell initially neighboring the wound, but then eventually it will leave the wound. So at the time, we didn't really know what the functions of these intercalations were, to be honest. No one had really seen these in a tissue repair context. And so again, we used mathematical modeling to try to address this issue. So in this case, we used a 2-dimensional vertex model. And hopefully you'll see that as the movie is playing, all else being equal in the parameters, but just in this one on the right, the wound is repairing faster because the intercalation rate we've set at a higher frequency. So essentially we call this a more fluid tissue. So the cells are able to flow into the wound edge much more efficiently. And Mike, the student who did the simulation, assures me he played them at the same rate. And you'll see that whereas this is finished wounding, this one repairing, sorry, this one is still struggling to do it. And eventually the pattern of the wound after repair is also different. So this is much more seamless, but this you can still see a scar at the wound site. Okay. And so again, simulations can predict in a way whatever it wants. It's only valid if we can really test it in vivo again. And again, of course, that's what we did. So we used many ways to try to change the fluidity of a tissue. But this one example, we knocked down myosin activity or reduced it slightly by driving rho kinase RNAi in the wing disc. And so this, we confirmed, makes the tissue more fluid. But also amazingly, the wound basically heals extremely quickly. And in fact, in half the time of a normal wound. So just by reducing tension in the system, this fluidizes the tissue and actually can seal the wound much faster. And so we're very excited by this work, and we're now trying to test this in sort of non-fly systems to see if this is valid elsewhere as well. But just in the last few minutes, I'd like to share some new data on tissue repair, which is to really look at this in 3 dimensions again. And so, as you saw in the first part of the talk, the wing disc is, of course, a highly 3-dimensional structure. And in fact, so is the wound. And so the fluidity work is all valid in 2 dimensions. But as you can see, the wound is actually itself a very tall, 3D structure. These epithelial cells are very tall, and the wound goes all the way through. So how does the tissue actually cope with this large 3-dimensional gap, and how does it seal itself in 3D? And so what Rob is seeing is that, in fact, as the wound closes in 2 dimensions, there are also complex 3D changes in the tissue. So looking apically using myosin, you can see that the apical surface essentially indents itself as the wound is closing. And in fact, so does the basal surface, which we label with collagen 4, looking at the basement membrane. So the surface also pinches in and as you see here, essentially shortening the cells. And so this is a hypothesis we currently have, which is that for very tall epithelium cells, one possible way to change, or to seal a wound would be just simply to change your height. And if you conserve volume, which is what we think does happen in the system, just simple geometry will ensure that by shortening, you'll increase your XY surface area. And this actually may be sufficient to, compensate for the lack of, for the discontinuity in the epithelium and actually seal the wound in itself. So this would contribute to a very fast and quick mechanism to wound closure. And so one prediction from this kind of logic would be that the bigger the wound, the deeper the wound would have to shorten itself in order to seal it, right? And that's indeed what we see, even within the small range. Obviously, if you have a huge wound, this wouldn't work, but within the range of wound sizes that we see, this is indeed the case. So the bigger the starting area, the deeper the wound gets as it heals itself. And so we are currently looking into a couple of questions, which is one, what actually drives this apical basal shortening of the cells? And two is, do these 3D cell shape changes actually contribute to wound closure at all? So, the hypothesis is great, but does it actually hold true? And so to answer the first part, what Rob is saying is that if you actually just go more basally, so apically we have the purse string, as many of you know, but basally into the system, we actually see these lateral actomyosin cables that line the sides of the wound. So you can see these cables here. Oops, it doesn't work if I scroll over. And so these structures basically extend along the lateral edges of the wound edge cells, just at the wound edge, all right? The rest of the cell doesn't really have these actomyosin cables laterally. And so specifically at wound edge, we see these structures. And so this, like whenever you see actomyosin structure, you think, oh, contractions, is it doing that? And so of course, like many of us, the first thing to try is to test, is there actually increased tension laterally in the system? And so what Rob did is to use laser ablation to ablate all of the lateral cables surrounding the wound edge. And then we track the apical position of the cells, looking at the myosin marker. And so when he ablates it, and if you follow the yellow dots, that's the original position, you'll see that upon ablation, the apical surface, essentially the height of the cell, starts to rise again and kicks up. And so obviously we can quantify this kind of change in height compared to, for example, control ablations. And so this change in height first shows us two things. First, that these cables are indeed under tension, but also these cables are really contributing to the cell shortening in the first place. And secondly, what about the wound area itself? And so when we actually perform the ablations, you'll see that this is the apical view. So looking at 2D surface area, and what we see is that indeed, after ablating these lateral cables, the area of the wound starts to increase again. So you'll see these quantifications. With ablation, the wound starts to increase in area again, whereas normally it should be continuously closing, reducing the wound size. And so what we'd like to propose is that, there are these complex 3D cell shape changes that occur driven by this increased lateral tension, shortening the cells, and this contributes to wound closure. And so there seems to be some kind of cooperativity clearly between, the lateral sides and the apical intercalations that we saw earlier. And so we're still really to understand this complex interplay between apical surface and lateral surfaces and how this cooperation helps to stabilize the wound area. And so this again is work in progress and unpublished, but we're hoping we can shed some light on this. And this could be very general mechanisms. And perhaps depending on the geometry of an epithelium, its height, its cell shape, the tissue will use different mechanisms to repair. And so again, we're very keen to compare and contrast different types of epithelia and see how these mechanisms would differ depending on the physical properties of an epithelium in the first place. And so in conclusion, I think I went through that very quickly. And in conclusion, what I've told you is really, the many ways in which a tissue can cope with mechanical stresses, right? And in fact, many of these mechanical forces are indeed very instrumental, especially during development, to really shape our organs. So for example, the forces that drive the 2D intercalations or growth, in fact, or oriented cell divisions and tissue folding. And so often if these persist for minutes or hours, our cells will respond and think, okay, this is really important instructive. So let's do what the tissue is trying to tell us to do, what the forces are trying to tell us to do, and get into shape. But of course, if these stresses are actually temporary, perhaps on the scales of a few minutes or even a few seconds, then the tissue has to respond using different ways to cope with that. So for example, the temporary kind of myosin alignments in response to the force, this would be a temporary way to resist any deformations. But if these forces then persist, then we'll move into this upper mechanism where the tissue will then respond. And so I think the timescales of these mechanical stresses are really, really critical for defining how a tissue responds to them. And so we're really interested in understanding that right now and how these timescales may change depending on the circumstances and the types of mechanical stresses that we actually induce to a particular system. And so with that, I'd like to end by thanking everyone in the lab. So I think I've highlighted the names of the people whose work I showed today throughout the talk, hopefully, or at least the names were in the corner. And then this is the current gang. And so this was our first party after 2 years of lockdown, so we're still masked up, and this is me here wearing my Phantom of the Opera mask, and if you can see, and so it's been really great to kind of start to go back to some degree of normality, and it's really great to be able to see everyone in live format. And I'd like to thank many collaborators who perform a lot of the theoretical work with us, and it's really a collaborative atmosphere that we have at UCL and also with our collaborators. And thank you to you for your attention and staying up so late.
Thank you very much. We probably have time for one question.
Great talk, thank you. Would you predict if you applied compression along the apical basal axis, would that accelerate wound closure?
It's really interesting you say that. We haven't done that consistently, but we've accidentally done that sometimes, just because when we image it or and it sort of sits against the cover slip, and for now we've kind of ignored that because it's something's gone wrong, but it does look like they wound differently. So I think we need to systematically investigate that. So that's something we really want to look at. So for now we haven't applied mechanical stretch or compression with our device to the wounding context, but what I would love to do is to look at, for example, if we apply external mechanical fluctuations, for example, we know that if we stretch it, we can change the mechanics. And does that then change wound healing responses? And that's really the future of the lab. We'd like to look into that. So yeah, great question.
Thank you very much. Let's take one from the chat. So in terms of adult morphology, can you say that in the wing, the growth is in the area of post-LM macrochetes is the slowest of what you see in the notum?
Sorry, so in terms of, just say that. So in terms of the macrochetes in the notum, whether that growth is the slowest in the notum part, we haven't actually noticed specifically, I have to admit. So it's not like, obviously the macrochetes have very precise patterns. And when we do our sort of mapping, we haven't noticed, obviously, the macrochetes popping up as specific patterns of different growth. So perhaps our data isn't enough detail in the notum section because we haven't focused specifically on the notum of the wing disc. But for now, we don't see there's any obvious change or difference, let's say, in those zones. Yeah.
All right. Once again, thank you very much.
Thank you.
Thank you. And our final speaker for tonight is Mark Ruiz from the University of Pittsburgh, and he'll talk about the origin of morphological novelties at cellular and regulatory levels.
Hey there, everybody. All right. First off, I just wanted to thank the organizers for giving me this wonderful opportunity to talk. I never thought from the first time I ever went to a FLY meeting that I would have the opportunity to speak in a plenary session. So it's like really special to me. And this is also like a really just amazing meeting. It's been so great to see everybody's faces in person. And yeah, so in my lab at the University of Pittsburgh, we are interested in the evolution of development, how morphology evolves, and in particular, we're interested in morphological novelties, structures that didn't exist before. How did they come about? How did the beetle's horn come about, or the turtle's shell, or the bird's feather, or these really crazy head ornaments in the tree hopper, for example, that just kind of boggle the mind in trying to imagine how they could have been genetically assembled or developmentally assembled at the cellular level. In a sense, what we want to know is, what is the origin story of these morphological novelties? And when we think of origin stories, I think one of the things that I find is a really interesting example of that origin story is superheroes. We want to know where our subject began. We want to know the starting point. We want to understand Peter Parker. And what happens to Peter Parker? We want to understand the change that caused Peter Parker to take on his amazing abilities. And of course, once that happens, once that change initiates, we want to understand what happens next as the amazing superhero emerges. And so our our interest in morphological novelties very much follows the same kind of story arc. Although I want to just let everybody in this room know that people in my lab don't sit around and talk about Spider-Man all day, just so you know. Okay, so we studied this problem from two complementary perspectives in my lab. We're interested in understanding how traits are regulated at the level of gene regulatory networks. This is a huge field of work in the evolutionary developmental biology discipline to, basically every single developmental structure that you're going to see is going to have a gene regulatory network that patterns gene expression to turn on specific subsets of genes in specific cells that then go on and do some sort of work to generate the morphological traits that we care about. A complementary approach, though, and clearly we could be studying those gene regulatory networks and asking what are the changes that are occurring within those gene regulatory networks. A complementary approach that we're interested in taking is really going from the bottom up, looking at the cells in these morphological structures and asking what exactly are these cells doing, and trying to understand at the cellular level what Peter Parker looks like, what Spider-Man looks like, or what the ancestral state looks like, and then the derived state of these morphological novelties. And so we could take a bottom-up approach to look at this from the cellular level. Now, of course, we would love to vertically integrate all of these levels of understanding and really try to figure out what are the genes that sit at the end of these gene regulatory networks that get activated in a cell type specific manner and yet directly influence the cellular behavior to generate these morphological novelties. And to be able to do this for something like the beetle's horn would be amazing, right? To be able to actually understand what are all the gene regulatory networks, what are those networks activating, and how are those networks activating specific effector genes that then cause something like the beetle's horn to come about. Now, of course, we can't do that as easily in a beetle's horn as we could do it in Drosophila. And so to study this problem, we have turned towards some of the most rapidly evolving traits that you're going to find in the animal kingdom, which are, of course, the reproductive structures. And so the Drosophila male genitalia is incredibly complex. It's a three-dimensional cuticle that has all sorts of projections that serve important roles in the mating process. In particular, we've been studying this epandrial posterior lobe. I'm going to call it the posterior lobe for the rest of my talk. It projects off this other cuticular plate called the lateral plate. And so I'll be saying posterior lobe and lateral plate a lot during my talk, and I apologize. This structure is incredibly diverse. Species that have a posterior lobe show a remarkable diversity. In fact, this is how we tell species of the melanogaster complex apart. Simulans has this kind of oven mitt-shaped posterior lobe, whereas Morchiana has this finger-like posterior lobe. Melanogaster, our home-based model organism that we all love and adore so much, has kind of a boxy-looking posterior lobe. And so it's a wonderful model to look at the differences in genetic control of how such a different morphological structure might come about. But of course, it's also a model for novelty because these are the only species that actually formally have one of these appendages. When we look at more distantly related species, they lack this structure. And so we can use melanogaster as a model system home base to study how the structure develops, how the structure is regulated at the gene regulatory network level, and compare these same things in species that lack a posterior lobe to start asking questions about the origin of this structure and how it came about at both the level of gene regulatory networks and cell biology. And so I'm going to start off from the bottom-up approach of looking at the cells in the posterior lobe to ask what they do. This is the work of a student who has since graduated from my lab. Her name is Sarah Smith. I'm very proud of her. She is doing a postdoc at Yale in Scott Hawley's lab. And she was really curious, how does the posterior lobe form at the cellular level? Well, coming into this, I'm not a cell biologist, and I apologize to all the cell biologists in the room for my complete and utter ignorance. But what we came to learn as we started working on genital morphogenesis is that, of course, the genitalia forms from an imaginal disc. It's an epithelial sheet that contorts into the shape of these genital structures in the choppy movie that you just saw. You can actually live image the genital disc. You basically cut a little window in the pupa, and it will basically, here we go. It will basically sculpt and elongate this posterior lobe structure into a flat epithelial sheet that then secretes a cuticle that then has its rigid characters that are used in mating. And so when we started working on this, we had no idea what was cellularly going on to make a posterior lobe. Is it cell division that's causing this? Is it cell movement that's causing this? And what Sarah found was really quite surprising to us. This is the structure that we can use to tell melanogaster from simulins from Mauritiana, that this structure is actually a single cell tall. When we look at clonally expressed fluorescent proteins in the cytoplasm, what we see is that those cells extend all the way from the basal lamina of this epithelium all the way up to the apical surface. So what you're looking at here is a single cell that's been labeled, and this is the basal lamina, that is the apical surface. So it's an extreme cell elongation process that contributes to the posterior lobe, and much the variation that we would see in posterior lobe morphology between species is going to be patterning which cells are going to become very tall and what is their arrangement. Now we didn't have many ideas for how this cell elongation might occur, but Sarah noticed something really odd in some of her antibody stains that she was doing. In fact, some of her antibody stains showed this non-specific staining above the surface of the epithelium. This is a place where we just thought there was nothing going on, but what she had seen was this diffuse material above the surface of the posterior lobe epithelium. It caused her to think maybe there's something going on in this place that we thought was just a void, an empty void where nothing was happening. And it turned out that work from Barry Thompson's lab and the late Susan Eaton's lab had shown how in wing disc and leg disc morphogenesis, the zona pellucida protein dumpy is deposited. It's an apical extracellular matrix protein, and it glues the wing blade to an overlying cuticle to help the wing maintain its shape. And so Sarah went looking at a YFP tagged dumpy line to see what does this look like in the genitalia? What does dumpy protein look like in the genitalia? And we were quite surprised and perplexed to see that dumpy is deposited as this giant gob of extracellular matrix that covers the central region of the genital disc. This is the region that covers the developing phallus, claspers, and it forms a network that connects directly to the posterior lobe. And so we have this complex network of extracellular matrix that connects this novel morphological structure to these other central fibers of dumpy that cover the whole genitalia. We're curious whether this might be important, and we did the experiment that I think everybody in this room would do. Is you order the RNAi and you cross it to the Gal4 and the right thing, you see what happens. And exactly what she saw was that there are huge defects when you knock down dumpy in the genitalia. So it's clear that these connections that this novel posterior lobe structure has to this vast network of extracellular matrix are important for the development of the structure, and therefore must have been important for the emergence of this structure during evolution. Now, to look at this in a comparative framework, we had to develop some lectin stains that would allow us to look at the glycoproteins of the extracellular matrix in both lobed and non-lobed species. And so she developed fluorescent lectin stains. In melanogaster, we got patterns that look very much like dumpy. So we see here in cross-section the fluorescent lectin binding to the posterior lobe, connecting over this structure that's adjacent, which is the clasper, over to this mass of extracellular matrix, which is connected to the phallus that's developing. When we looked in a non-lobe species, Drosophila biarmapes, what we see is that this lectin labels quite a bit of extracellular matrix. Indeed, what we see is that over the central phallus of the species, there is, again, this giant mass and network of extracellular matrix. It covers the adjacent clasper structure. And yet, where a cell would normally be making a posterior lobe if it was melanogaster, this connection is not nearly as strong as the connections of extracellular matrix you see in Drosophila melanogaster. And so this data really suggests two things that are critical. First off, it would seem that something is critical to genital development, just ancestrally, that all of these species are using this extracellular matrix mechanism for something. And the second bit is that in Drosophila melanogaster, which has evolved this novel structure, there has been a repurposing of that ancestral matrix, basically redistributing it to connect it to these new cells. And very much in line with this hypothesis, we see that dumpy mRNA is expressed in a pattern that correlates with the posterior lobe, and species that lack lobes show less dumpy expression there. Indeed, a postdoc in the lab, Ben Vincent, has shown that, and he had a nice talk the other day and a poster in the poster session, that Drosophila mortiana, which has a small lobe, differs greatly in dumpy expression from Drosophila simulans, in which this lateral plate basically makes one giant metal lobe. And so we see differences in dumpy expression that correlate very well with this. Okay, so from the data that I've shown thus far, we can say that there is this ancestral Peter Parker, if you will, mass of extracellular matrix associated with the central phallus and clasper that has then expanded out to cells more laterally that then make posterior lobes. And this must have been important for the evolution of this structure. This raises two questions, though. It raises way more than two questions, but it raises two questions that I'm going to talk about today. And those two questions are, well, what regulates these differences in apical extracellular matrix deposition? And what on earth is this ancestral gob of dumpy that is on the developing genitalia doing? Our work on the gene regulatory network side sheds some light on what might be happening. So I'm going to be presenting work from some additional graduate students in the lab. This started with my first graduate student, Bill Glassford, who gave a great talk yesterday. He's in Richard Mann's lab studying Hox proteins. And one of the first undergrads that I had in the lab, we basically went to my colleague, Jared Campbell, who's a developmental biologist who works on leg discs and wing discs. And we said, give me all your antibodies and we're going to throw them on the genitalia and we're going to find genes that are patterned in this posterior lobe structure that we're interested in. And from this, Chaz and Bill found that the Notch ligand delta is patterned right next to where the posterior lobe forms, suggesting that Notch delta signaling might be important for posterior lobe development. Indeed, when we look in species that lack posterior lobes, we see that delta is present in a similar location, although in a much less expanded pattern, suggesting that maybe patterning differences in Notch and delta are important. We went in to manipulate Notch delta signaling, and indeed, when we knock down delta with a lobe-specific driver, the posterior lobe is greatly reduced. So it's clear that this Notch delta signaling is important. Similarly, if we overexpress the intracellular domain of Notch in this same location, we can make a ginormous posterior lobe. And so it's very clear that Notch delta must be very important for tweaking posterior lobe shape and size. And so this motivated us to do kind of a painful experiment, which is something that my lab does a lot of, which is to look for enhancers. So we wanted to find the regulatory elements that drive the expression of delta in this pattern. And so several years of work went into making little reporter constructs around the delta gene. It's a big gene, it's kind of a pain to work with, And this was started by Bill and is being finished by a senior graduate student in the lab, Donia showed you. And so we found two enhancers that together nicely recapitulate the pattern of delta expression in the genitalia. Now, Donia went on to ask questions about whether this enhancer has evolved. And I'm just going to make a long story short and say that we think that changes upstream of this enhancer are responsible for any kinds of expansion that we might see. But Donia noticed something really interesting when looking at these enhancers. Tracing the timing on which they begin expression revealed something very odd. And to describe this to you kind of more fully, I actually have to show you a little bit of the really confusing world of genital disc eversion. So the genital disc begins as like a deflated basketball. It's basically kind of crunched in on itself, and it's inside out. The basal lamina is on the outside, and all the apical surfaces are on the inside. And over the first day of development, the genital disc basically turns inside out. The anal plate extrudes out first, and then the lateral plates and claspers and all the structures with the posterior lobes on them extrude out over that next day of development. And so Donia was looking at her enhancers, asking when do they first activate? And what she found is that no matter how early she looked, this enhancer is active. So this is 24 hours, and we can see a nice pattern that looks exactly like the delta expression in these cells. This is a 5-hour after puparium formation disc. The anal plate is just kind of starting to pop out from the genital disc, but there's our little cell population. It's on the inside of the genital disc. And when we look in a third instar larva, this expression pattern still exists. And so the significance of this is that this is basically a week before there ever is a posterior lobe, marking the cell population in the genital disc where the posterior lobe might form. And indeed, Donia has gone on to show that this pattern is exactly where delta is expressed in both lobe and non-lobe species. So we are convinced that these are real patterns of delta, ancestral, Peter Parker, if you will, to the expression, ancestral to the emergence of the posterior lobe during evolution. And so this raises a very interesting question, right? Why on earth is Notch delta signaling turned on a week before there's a posterior lobe in this patch of cells that then go on in non-lobe species to basically be expressed in a place near where the lobe would form, but is expressed in a slightly expanded pattern in species that make a lobe where it seems to be important for generating a posterior lobe. It's important for patterning something about the posterior lobe. So Donia set out to figure out, well, what is the significance of this pattern? What does this pattern do? She looked through tons and tons, and this was all in the pandemic. It was crappy. We all hated it. She went looking for drivers that could knock down delta in these patterns, in this pattern in the early larval genital disc that could tell us something about what happens. And so here's a close-up of the disc showing delta expression, and when she knocks it down with one of the drivers that we found, we very specifically affect this little lateral patch of delta. And so what happens? What happens to development? We thought, okay, well, this is just going to change all the patterning. This is just like some early patterning thing. It's Notch delta. It's going to like do everything. It's right. It's Notch. The thing that's really weird about this is that when we knock down delta in this patch of cells, what she finds is that 100% of the time, the pupa fail to avert properly. But basically, we get this phenotype at one day of pupal development, where the only thing, if you see a genitalia poking out at all, you actually have quite a few where they just don't avert at all. You see just the anal plate poking out of where the genital disc is. And so this would suggest that this signaling center that 2 days into, or a day or two into genital development during pupal development is critical for patterning the posterior lobe, is involved in this aversion process, and it's doing something important there, which to us was kind of unexpected. I mean, Donia thought maybe this was going to be the case, and I was like, no, but I always love it when a student proves me wrong. Yeah. So Donia really wanted to kind of connect this to what we know about posterior lobe development. And she got really curious about whether this might have something to do, whether there's a connection between dumpy in the larval genital disc and dumpy that is occurring during pupal development of the posterior lobe. And in fact, when she looks at the third instar genital disc, what she sees is that, remember, the genital disc is like this deflated basketball where all the apical surfaces are kind of scrunched up against each other inside the basketball. What she sees is that there's actually dumpy along the inner surface where these apical surfaces meet, and including the region where delta is expressed. And indeed, as the genital disc is everting, we see connections of dumpy into from the place where the posterior lobe forms that extend all the way inside. When we knock down delta in this larval genital disc, what Donia found, and this is kind of hot off the presses data, but I really wanted to show it, is that these connections don't exist between the cells that are next to where the posterior lobe forms and the inner cells that then are getting pulled out by some mechanism. And so what this suggests is that perhaps dumpy is important for the eversion process, that basically as the cells of the anal plate are coming out from the inside of the genital disc, that these connections with dumpy might be important for dragging along the rest of the genitalia. That's just an idea right now, but it's something that we're very excited about. I wanted to kind of take it back to Spider-Man because it's like my favorite thing. I've been so proud of it. I've been telling my friends, I'm going to talk about Spider-Man during my talk. So one of the things that I think a lot of us find compelling about superhero stories is that we see a little bit of ourselves in Peter Parker. I do, I know. And I think it's important to remember that Spider-Man would never exist without Peter Parker. We're all Peter Parker at some level. And we're all important, we're all unique, we all have, everybody in this room has incredible insights, we're all scientists. And I don't know who needs to hear that, but I just wanted to say that for y'all. Much like there's no Spider-Man without Peter Parker, there's no novelty without some pre-existing complexity that exists within these structures. And that's what we've been studying. That's I think the message for us is that these novel morphological structures are built in the context of already incredibly complex developmental processes. And by studying what is happening in the before state and what's happening in the after state, we can really start to learn with nuance how what the seemingly insurmountable developmental processes might evolve by slight adjustments of that pre-existing complexity. That is, we see this developmental signaling center that well predates the origin of the posterior lobe. It seems to involve these connections with apical ECM, providing this context in which you could expand the apical ECM and provide a surface upon which something as unique as the posterior lobe might evolve. Now we have a lot to figure out with this system, but it definitely shows just how complex a system might be before one of these novelties evolve. With that, it's my story, I'm sticking to it. I need to thank my lab. I have an amazing lab. I'm just so proud of all of my trainees, the trainees that came here. Just wanted to make a big shout out for Yvonne Mendez, who's a senior graduate student. He's looking for a postdoc. Ben Vincent gave a great talk and poster, did some of this dumpy stuff with Simulins and Mauritiana. Gavin Rice has been a huge member of the GSA community. He's been volunteering for all sorts of stuff in the GSA. Yeah, so definitely, I hope you all meet my trainees. I'm really proud of them. Thank you to my collaborators and funding sources and all that. I'm happy to take your questions. Thank you.
We do have time for questions.
I talked really fast.
Hey, Mark, this is Deepika, also at the University of Pittsburgh.
Hi, Deepika.
Hi.
Good to see you. How you been?
Good to see you. I've been well, trying to stay awake. I'm trying to understand this from kind of a naive biologist point of view of the Darwin finches, right? So you see all these various different shapes of genitalia, but what is kind of the purpose? Like, what is kind of the selection pressure that drives the evolution of these,
Yeah. No, that's a great question. So, we really don't know. We know that you have to have them to mate, and we know that there's all sorts of conflict that's probably going on and driving them, but the specifics of why these are taking on their specific changes. My favorite model is that female choice is changing and that the males have to kind of keep up. And that's, part of that arms race that's going on between morphology and behavior.
Hi, I'm Oğuz Kanca from Baylor College of Medicine. Is it my impression, or in the genetic manipulations where you had the smaller posterior lobe, you had more bristles in the genitalia? Is this a misdifferentiation of the epithelium into bristles, or?
My assumption there is that notch signaling is important, incredibly important for lateral inhibition, and that by any manipulation that we use that would affect notch signaling in that tissue is also going to affect that lateral inhibition process.
But I think you had it with dumpy knockdown as well, not only with the delta.
Oh. I don't remember seeing that with the dumpy, but we do know that some of these extracellular matrices are in the bristles, but why would you get more bristles? Yeah, it's possible that dumpy is affecting the signaling around those bristle precursors or something like that. Yeah, really cool point.
Might be that you have the same number of bristles compressed into a small area because it doesn't expand.
Potentially, yeah. No, it could really be something like that, yeah.
I have another question. I'm going to ask you to speculate. So
Oh, no.
with this expansion of the posterior lobe, do you think that there is some pulling force that's being transmitted to those elongating cells from a different part of the genitalia via the extracellular matrix? And if yes, what do you think might be applying that force?
Yeah, I think it's possible that we really don't know biophysically what's happening here with this matrix, but we do see that it shrinks over time, like late into pupil development, the actual area that it takes up shrinks. And so it could be that something is causing it to contract just overall. We don't necessarily think that any one thing is pulling on it, that there's a puller that then drags these things along, at least in the context of the posterior lobe. But it might be just that it's a surface upon which these cells are crawling and differentially adhering to as well. So,
well, thank you very much, Mark.
Thank you. Thank you so much.
Well, and that concludes our session. Thank you very much to all the speakers for excellent talks. And I would tell you to go and get some sleep, but I suspect that's not what you're going to do.
I know, I'm really tired too, but
That was a long, long time ago.